The Regeneron Genetics Center aims to identify novel drug targets using high-throughput next-generation sequencing. Over the last decade, our collaborations with major healthcare systems, academic institutions, and Regeneron clinical trials have enabled us to sequence over 2.5 million human exomes. Let's learn about the steps it takes to sequence DNA. Before DNA samples can be sequenced, they must first undergo several preparatory steps. The first step of our sequencing process is sample receipt and normalization, and involves the careful handling of genomic DNA samples. Collaborators send in already extracted genomic DNA samples in 96-well tube plates. We use a buffer solution to normalize the DNA to ensure consistent concentration, and then transfer each sample to new 96-well plates. With the samples prepared, we move to the second step, Precapture library preparation. At this stage, DNA undergoes a series of modifications to ensure its compatibility with the sequencing platform. Initially, the DNA is fragmented into precisely sized pieces, then short, specialized DNA sequences, known as adapters, are attached to the fragmented DNA ends. After that, the DNA fragments are amplified through PCR, or polymerase chain reaction, and during this phase, molecular barcodes are introduced to each sample for tracking. Next, we conduct normalization and pooling. This step ensures uniform concentration across all DNA libraries. This is important because these libraries are then pooled together, and we need to prevent individual samples from dominating the results. The barcodes that were previously added enable us to distinguish between the different samples post-sequencing. For step 4, we move to exome capture. Our goal is to selectively enrich the exome, the protein coding fraction of the genome that is most critical for understanding gene function and disease association. To do this, we add exome oligo probes to the pooled libraries, which bind to the exon regions in each patient's DNA. Next, the intronic regions are washed away and discarded to effectively isolate the exome from the rest of the genome. Following enrichment, the exome DNA undergoes another amplification step using PCR to ensure we have enough material for sequencing. Now, the DNA is finally ready for step 5, sequencing. Here, we normalize and pool DNA libraries a final time so that we can maximize the efficiency of our sequencing runs. We are sequencing hundreds of patients all at the same time. The DNA, a flow cell, and a set of reagents that come in pre-filled cartridges are loaded into one of our NovaSeq X Plus sequencing instruments, and a run is started. The DNA sequencing method that's used is called sequencing by synthesis, and a run takes about a day to complete. At the end of the run, files are created for downstream genetic analysis.